Hello friends, welcome to With My Plant. Today we are going to understand the polar transport of auxin. Actually, this polar transport is actually responsible for the apical dominance in the plant. And this class will be coming under auxin series. Okay, let's understand the polar transport of auxin. Here in this picture, I have presented the numbers and in the right hand side I have enlisted the action. Okay, so first if we see this picture, whole picture, this is the arrangement of cell in plant and or in stem, you can say, and as marked here, the this is the cell wall and this is the plasma membrane or cell membrane, you can say. And this is the apoplastic region outside the cell. This part is the apoplastic region, and the direction of the auxin transport is from up to down, up to down. Okay, so in the first point, as I have marked here, the first point, the what is happening in, as per chemiosmotic hypothesis, the gradient of pH between cell wall and cytoplasm drives a IAA accumulation. So here you can see the pH inside the cell is 7.0, sorry, 7.0, whereas in the apoplastic region it is 5. So there is a difference, means inside the cell there is basic pH, and in apoplastic region it is acidic pH. Because of this acidic pH there is accumulation of IAA, auxin. Okay, so because of this difference, or inside it is a basic pH. So the IAH, that is the IAA minus plus H plus. Okay, so it will be entering into the cell, and through auxin influx carrier. Okay, influx carrier can be your aux1. Okay, through that auxin is entering into the cell in the form of IAAH. It can be entered in the form of IAA minus through simple diffusion. Okay, now IAH, when entering into the cytoplasm, due to this pH difference, it will be deprotonated. That means it will be separated as IAA minus and H plus. Now this IAA minus cannot um, go outside the cell or to the apoplastic region if there is no efflux transporter or efflux carrier. So these efflux carrier uh, are distributed ununiformly or asymmetrically in the cell, and they are mostly present in the downward direction. That's why there is a polar movement. The sometime question came, who is the responsible molecule or responsible protein or responsible carrier for polar transport? The answer will be this efflux carrier. These efflux carriers are present in the down direction or in the lower direction. That's why the movement of auxin is from up to down. Okay, so what is happening in the first point, I will read this. Chemiosmotic, because of this chemiosmotic hypothesis, which is saying that pH gradient between cell wall and cytoplasm drive IAA accumulation. Cytoplasmic pH, as it is more basic, leads to deprotonation of majority of IAH and trapping inside the cell. Now IAA minus can exit only through active efflux mediated specific carrier, that is the efflux carrier. Now asymmetric distribution of efflux carrier promotes your polar auxin transport between the cell. So Yes, this is very important. These efflux carriers are mostly pin proteins from multi-component complex for auxin efflux, along with another category of protein, that is NBP, and also some unstable components. Okay, so aux1 is basically a influx carrier, and apart from aux and pin, there are other families such as pgp family and abc transporter are responsible for this efflux and influx of your auxin. So pin protein aux1 is basically auxin 1, and also another one is lax, that is like auxin 1 genes. Then another group, as I told, there is PGP. The full form is poly G glycoprotein subfamily, and another is NPA binding protein, that is called NBP, and another I told ABC. It is ATP binding cassets. So this is all about the process of auxin transport or polar auxin transport. Let's revise it. So auxin is when there is difference in cytoplasmic and apoplastic pH. Um there is accumulation of auxin, and it is basically in the form of IAAH. But IAH, when it is getting transported through membrane, it needs one influx carrier. But when it is in separated form, that is as IAA minus and H plus, it can be moved through simple diffusion. uh, because of this basic pH inside the cell, thus IAA cannot be effluxed through simple diffusion um, or other way. It needs one efflux carrier, and that efflux carrier is actually responsible for the polar transport, as it is asymmetrically distributed throughout the cell or cell membrane, and it is mostly present in the lower side of the cell. That's why the auxin is moving from up to down. So this is all about, and these yes, this efflux carrier protein is basically pin and nbp. So this is all about the polar transport of auxin. I hope you guys understood the transport system of auxin, and because of this polar transport of auxin, there is apical dominance in the plant. So I hope you guys understood. If you are not understanding, you can comment below, and I will try to make you understand in a simpler way. Thank you.